Hello, my name is Breck Torgerson, and my topic is genetically modified organisms, commonly known as GMOs. So what are GMOs? They are classified as living organisms that have their genetic material artificially changed in a laboratory through genetic engineering. And the examples we'll be going over today are glow-in-the-dark mice and worm-defiant cotton. To begin, GMO animals are used for two different purposes. One is to improve the consumption and production needed from the animal, and the second purpose is to be used as a model within a laboratory. This means they are used to study the function of certain genes and how they relate to the overall health of the organism. Mice are a common model within the research of GMOs. In the case of these mice, they have been injected with genes from fireflies to glow in the dark, more specifically to glow when tumor and cancer cells are present. Led by lead researcher Timothy Blake, he and his research team wanted to see the probability of gene therapy and its possible techniques for vaccine and cancer treatments. These type of experiments allow scientists to follow and understand how genes can be introduced into another organism in hopes of benefiting the organism and eventually mankind. So how did the scientists create glow-in-the-dark mice? They took the RNA copy of the gene responsible for glowing in fireflies and inserted it into the mouse's cells. The scientists specifically targeted the p16 gene, which is a gene responsible for aging and cancer suppression. But the biggest problem the gene therapy had was that the negatively charged modified RNA had a difficult time going through the positively charged cell membranes. To combat this, researchers created what they called a charge-alternating releasable transporter, or CARTs for short. These transporters are designed to change the polycations to neutral small molecules that now allow RNA an easier way to get into the cell. And once in the cell, the CART will disintegrate and release the RNA into the cell membrane. With this, scientists were able to document results showing how p16 gene combined with the firefly gene detected tumors before they even began to show visibly on the mice. It also showed that older mice glowed a lot more in comparison to its younger counterparts. Our next example is that of cotton. In the beginning of the production of GMOs, GMO plants have been consumed since the mid-1990s. Many GMO plants have been modified to benefit both the farmer and the consumer by extending the shelf life and having the ability to ward off potential diseases and parasites. The overall crop yield and the end-of-the-year discardment have increased and decreased respectively due to the modification of cotton. In the case of cotton, they were genetically engineered to kill bollworms. Bollworms have been the biggest devastator to cotton crops in Alabama and Asia for the last several years. The larvae chew holes into the bulbs of the cotton, effectively eating it from the inside out, rendering it useless for production. By genetically modifying the cotton, it not only protects the plants from the bollworms, but also lowers the exposure to dangerous pesticides on the crop and surrounding areas. GMO cotton is engineered with the gene that codes for toxin crystals in the cry group of the exotoxin that is from the Bacillus Thuringiensis. This bacteria naturally lives within soil, and the proteins that it produces is very toxic to insects, such as bollworms. These proteins, however, are not toxic to humans and animals. By placing the toxin gene within the cotton plant's genome, it naturally begins to produce these proteins. So how does the cry toxin work against the worms? When ingested, the crystal toxin begins to break down the midgut cells by inserting into the target membrane and forming pores. These pores produce even more toxin crystals. The toxins then break down the cell's proteins into smaller peptides activated by the midgut proteases, which are the enzymes that break down proteins. And then it binds to specific receptors located in this insect cell membranes, leading to the eventual death of the insect. They can travel across membranes with ease due to the toxin's classification as a pore-forming toxin, or a PFT, which is a class known for the ability as a water-soluble protein that can change its shape to cross any membrane of the host. To recap, we learned that scientists have figured out how to take an RNA copy of a gene responsible for glowing in fireflies and insert it into a mouse's cell membrane, resulting in them glowing. They can do this with the help of charge alternating transporters to help the negative RNA pass through the positive membrane. We also learned that GMO cotton has toxin crystals that use spores to reproduce and break down important proteins in insects like bollworms to effectively kill them without hurting the cotton and the environment in the surrounding area. Thank you for listening to my presentation and have a nice day.